Welcome back for the keynote session. And after your caffeine shot, I hope you're ready for some dopamine because the next speaker is as such. And it's my pleasure to welcome Jack Gilbert. Professor Jack Gilbert is from University of California, San Diego, professor of pediatrics, as well as from the Scripps Institute of Oceanography. And Jack's the co-founder of the Earth Microbiome, the American Gut Project, among many other exciting projects, including hospital microbiome, home microbiome, and the list goes on. So, yeah, my association with Jack basically stems from the fact that I was initially part of the Earth Microbiome Project during my time at the Rob Knight Lab. And I'm here as a representative of Reckitt Health. So this is part of the industry. And thank you guys for putting up a great meeting, and a shout out to the NIST team. And Jack, take it over. Looking forward to your talk. Thanks.
Thank you. Thank you. I appreciate it. I'm assuming everyone can hear me okay?
Yes, we can.
Good. Just checking. So yeah, it's my great pleasure to give this presentation. It's a really interesting one. As I think I've heard from a number of the speakers, talking about standards can often be a little bit dull. And I've dealt with that for 15, 20 years of my career now. And to me, it's anything but dull, but it's become an essential part of our ability to do massive scale microbiome studies more generally, and is an ongoing problem. And so I'm going to divide my talk a little bit between overviewing how some of the existing standards have enabled us to do some large-scale studies, and then also to highlight what I think, what myself and my colleagues, hopefully some of you are in the audience I know very, very well, will agree are some of the exciting opportunities and some of the hurdles that need to be dealt with. So I do have disclosures, tend to work a lot with industry, and I'm very interested in entrepreneurship. So I like to start most of my talks with this figure. I've probably been giving this slide for 15 years now, but it really deals with the idea that we're dealing with an incredibly complex, multi-dimensional, highly dynamic system that's under constant change. And I know Li-Ping said earlier that we may not want to standardize everything to understand it, and that's very, very salient and very important detail. We can't standardize everything to deal with it. All we can do is really try and come up with approaches that allow us to ask specific questions. And in that instance, it may be necessary to standardize elements, but when we ask different questions, we want different processes. So there is and should never be no one-size-fits-all solution for all the microbial communities on planet Earth. My interest in standards started when I started my first independent position back in 2005, working for the Plymouth Marine Laboratory back in England, where I grew up and obviously established myself a little bit. And that was a private company, and I was approached by a fantastic researcher slash social influencer of the time, Dawn Field, who said she was really looking for scientists who were generating really big data sets with the understanding that the contextual information surrounding those data sets needed to be recorded in a way that could be uploaded into the INSDC, so that's GenBank, the EBI, and the DDBJ, to provide everybody with the opportunity to be able to reanalyze those data sets. Later on, working with Casey Green, we started to refer to these people reanalyzing data sets as data parasites in a really positive way. Somebody called us data parasites, so we took that moniker and owned it and said, yes, we're data parasites. But at the time, I was generating a lot of data for a study in the English Channel. And Dawn Field was the person that really saw the value of this and helped to bring together the entire community around the Genomic Standards Consortium. The logo of the Genomic Standards Consortium is very, very similar to the Earth Microbiome Project logo, which I'll show you a bit later. This is because a brilliant designer, Owen, who was working with us in the GSC, designed this and then designed the EMP logo just afterwards. So anyway, I'll talk about that in a bit. So unfortunately, we published two papers out of the Genomic Standards Consortium that really changed the field. The first one in 2008 on the minimum information about a genome sequence, And then the second one, my marks, which became the MIXS specifications. And this really was led by Dawn and to a certain extent Peter Sturck, both of whom unfortunately passed in the last 5 years. And both of whom were very close friends of mine and my family. And Dawn was a fantastic supporter of my research, especially when I was an early career PI, and also a great mentor and friend. So very sad that they've left us. But this is Dawn actually. I accidentally caught this image. So I thought it was really cool when she loved this one of her holding her son on a walk we did in Devon. But obviously it's just an optical illusion, but it's a fantastic optical illusion. Anyway, so both of them helped to really drive this field forward and set the standards, if you will, pun intended, for implementing standardization of data reporting in microbiome studies, which led us to some really incredible opportunities in scientific discovery. So the MIGS, MIMS, so MIGS is minimal information about a genome sequence, MIMS is minimal information about a metagenomic sequence, which I helped to develop, and then my marks, which is about marker gene sequence databases, so 16S, 18S, ITS, and then we've even got new checklists, which we started to develop around pan-genomes, all provide a way of recording data in an effective way, so that if you do use different approaches, different DNA extraction approaches, different sequencing methodologies, different bioinformatic analyses, all that information can be captured in the data that you actually release to the community, so that data parasites like myself and Katie Green can come in and use that effectively and not have to bug you consistently to be able to access that information. So while this isn't necessarily about standardizing the approach, I'll get to that later, it is about standardizing recording of the approach, which, as I know, this team of people that organize this meeting is very, very passionate about. And it's something that I'm really glad to see is continuing. So I wanted to start out about what drove this forward. We started working on standards in order to capture information about a particular location in the world, the English Channel. I like to think of the English Channel as the rectum of Europe. It's the colon, if you will. The water from the North Atlantic pummels through here and then is pooped out this little sphincter. I heard that we did like the poop emojis, so I can make poop analogies as well. It comes out of here and is pumped out into the North Sea. So this is an area of water that has been studied for about 150 years with near constant, other than world wars, near constant weekly assessment of the biology and the physicochemical properties. It's an incredibly rich database that contains huge amounts of data about many different factorial variables. And what we did in 2005 when I arrived there, using my startup, was kickstart the analysis of the microbiome. This just so happened to coincide with the development and implementation of long-read sequencing, next generation sequencing technology, such as the 454 Roche Pyro sequencer, which we were one of the first people to deploy in Europe, especially, but even in the world, into a large study design where we applied metagenomics and metatranscriptomic analysis in 2005, 2006, 2007, to understand the microbial dynamics of this system in the context of this wider physical environment. So this was our first attempt. We started to use amplicon pyro sequencing. First of all, we had one site, we had 6 years of observations and 72 samples, and it took us about 7 years to get that published. So we were determining how the microbial community structure varied in the English Channel. We collected all of this environmental parameter metadata and placed it in the minimal information about a metagenomic sequence standard language, and uploaded all that information into GenBank. And this got the attention of a number of different groups, including well, I'll talk about in a second, the International Census of Marine Life, microbiological life, which was led predominantly by Julie Huber, who's a fantastic and groundbreaking researcher from the Woods Hole Oceanographic Institute, previously of the NBL, and other team members. But this study helped us to place it in a global context, which I'll mention in a second. But here, every single one of these little pies, if you will, is a month, and we had 10,000 16S frees from every single one of these months over this 6-year time frame. The reason I'm showing you that is because we wanted to understand a phenomenon that was associated with microbiome analysis, whereby when you're looking at amplicon sequencing data, you're dealing with proportional data, and you're dealing with the fact that when something becomes abundant in that database, it takes over all of our proportional read space, so that a single organism can make it look like this is a very undiverse system. And our system was diverse, but it fluctuated in diversity. So the y-axis here is just the log of diversity, and the x-axis is time. And so over the years, you get these fluctuations up and down, with richness of the number of species peaking in December and troughing around June. And we wanted to understand if this was a phenomenon of organisms being introduced into the environment and continually turning over. Because remember, this is the colon of Europe, right? So all the water's being pushed through. The residence time of a single, say, square meter of water is only about a week. It hangs around for a week and then it's flushed out. And so we wanted to know if the bacteria were changing because they were constantly being fluctuated, or whether they were just adapting to these constant changing environmental conditions. And so to understand that, we worked with the International Census of Marine Microbes to analyze one of these time points, and we randomly picked December 2007 to analyze 10 million. Sounds huge, right? This was crazy at the time. 10 million sequence reads from that one time point. What we identified is that all of the 16S amplicon units, operational taxonomic units at the time, OTUs, were present in this one time point. So at any point, any of these previous months or subsequent months, any of the samples that we saw in those 10,000 reads per sample were nearly all present in December 2007. And because all this data was standardized with the IMMS and MyMarks standards, we were able to compare our data to the International Census of Marine Microbes continual data set. And this included sample collections from subtidal rocky reefs, oceanographic environments in the bathypelagic or the abyssopelagic, mangrove swamps, estuarine systems, abyssal zones, sediments, even cold seeps and hydrothermal vents. And in this, we demonstrated that the bacteria found at each one of these locations, 50% of all of them could be found in that one time point in the English Channel, which to me suggested that the English Channel, being English, was the most important place on Earth for microbiology. Or, ego aside, it's possible that most of the microbes are just present in all environments and just fluctuating in and out of proportional abundance in our data sets based upon the coverage and their ability to thrive and survive in that environment. This references Bass-Becking's hypothesis, right? The idea that everything is everywhere but the environment selects. But without standardizing all of the metadata and all of the microbiome data in this context, we wouldn't have been able to make these comparisons. And this study design really led us to launch the Earth Microbiome Project in around 2010. This is an infographic on the left from the first paper, which was published in 2017. I say the first paper, data paper, because there are more coming. We have one that I think has been accepted and should come out soon, which I'll mention briefly. But this was an effort to catalogue the microbial diversity of the whole planet using the Genomic Standards Consortium standard languages developed by Dornfield, Peter Sturck and colleagues, that enabled us to take all of this data from hundreds of different hypothesis-driven studies and place them all together in one amorphous data set, right? And to therefore ask interesting questions like, if we can standardize all of the information, can we understand how microbes vary across pH range, or between different animal species, or across different types of environmental features? And so this study, which has now, I think, over 240 different peer-reviewed publications that it has enabled, was a hypothesis-driven study of hypothesis-driven studies. So hypothesis is within hypothesi. It's kind of crazy. But it facilitated us to start to do a number of things. And this is where we switch from data standards to methodological standards. Because the only way in which this study could be facilitated effectively was by standardizing our DNA extraction protocols, our amplicon and metagenomic sequencing protocols, our metabolomics protocols, in a way that allowed us to effectively compare between samples. And a lot of this methodological standardization was led predominantly through Professor Rob Knight's lab, who also works here at UCSD now, but previously at University of Colorado, and then my lab and various other labs around the country, to enable us to create something which was validated across hundreds of environments and could be deployed effectively and updated when necessary. And a good example of this is that when we're analyzing different environments, different types of ecosystem, like animal body, plant bodies, secretions from animals, all the way through to water that's non-saline or saline, we found that the same DNA extraction protocol and the same PCR primers, for example, for the 16S analysis, acted differently on different taxa under different environmental conditions. So our attempt to standardize everything was biasing everything. And that's a really important point to understand. Every time we standardize something, every time we analyze something, we're not analyzing it perfectly, it's all a bias, right? So really driving this home and trying to understand it effectively was something we were super interested in. Yep, just checking. Okay, so we found using this large-scale data really interesting things like this concept of microbial osmosis. Like the Bass-Becking idea of the English Channel being the most diverse place on Earth, obviously it isn't, we found that really diverse locations, like up here on the right, so the y-axis here is the number of different phyla that we could identify in a sample, and then I've just taken 2,000 randomly collected samples from the Earth Microbiome Database, and I've ranked them based upon diverse on the right, or very low diversity on the left. So, for example, over here we've got samples that have just got 2 different phyla in them. But what we can always find is that really diverse environments capture all of the diversity of the system in a low diverse environment. So the low diverse environment, as Bass-Becking would have predicted, is just selecting from this morass of microbial bombardment that's constantly going on. And so you can see that when you break it down into low or high definition structures. So up here on the top, I've colored everything by sample type. You can see the key at the bottom, but every single bar here is a genus. And then on the bottom, I've broken those genera into individual strains. So they're not directly proportionally representative, but you see that genera are found genetically distributed across myriad environments. So if I just take this on the left, I've got samples in non-saline aerosols, and in animal distal guts in brown, and then in animal proximal guts, and then on the surface of animals, and then also in water, both saline and non-saline. So these genera can be found distributed over a huge distance. Evolutionary speaking, they've adapted via their species and subspecies modifications. And when we look at the strain levels, we see much greater selection pressure. So some of these strains are only found in non-saline sediments over here on the right in pink. And then this dark green in the middle is some bacteria that are pretty much only found either in the soil or in the plant rhizosphere, which is just selected soil. So these, when we get down to strain level or genotype level adaptations, you see those occurring. Again, because we could standardize the methodologies and the metadata, we can make these very broad level comparisons and identify strains which are universally distributed or which are highly selected. And so this has led to dozens of papers focusing on deliberately driving both the methodological standardization. So, for example, here, ultra high throughput of microbial community analysis. This short communication from ISME journal in 2012 is still the most cited article in the ISME journal. It's a ridiculously highly cited article. And then, things like the Blur analysis or Emperor or the MyMarks Nature Biotechnology paper I mentioned earlier, or the Biome Matrix. These approaches have helped us to validate and standardize our analysis over broad scales, create a data analysis ecosystem to deal with diverse data, which is standardized under appropriate validated protocols. So the EMP currently, the Earth Microbiome Project, has sequenced 200,000 samples. We've done them all with ADCON sequencing. We have some with metagenomics, some with metatranscriptomics and metabolomics, and we've done them from around the world. We have also our Microsetta initiative, which has a collection of multiple human and some environmental studies, one of which is the American gut, which has processed 30,000 human fecal samples. So at least in our data collection, I can say, which we hold in Cheetah, QIITA, we have over 500,000, so 500,000 samples that have been processed and uploaded and available in that system. It's an enormous amount of data, and nearly all of it has been used, the genomic standards protocols for data standardization and the EMP protocols for methodological standardization. But we still can't design build a microbiome that works for something we want, right? We can't even predict what will happen to a microbiome in an environment or in our bodies. We still don't know that, right? Even with an extreme diet intervention in a person, let's say I take a person and I feed them only Milky Way bars, right? So that's all they're eating for like 24 hours. I can't predict what would happen. I can observe it, but I still can't predict it. So we've done an enormous amount of sampling, wonderful standardization, but we're still not able to go and take that information and predict what's happening. So let's talk about what we see as some of these problems, right? We must care about bias. That's number one. So every method is biased, right? The field historically hasn't cared. I haven't really worried about it. I've focused on getting my graduate students to process their data about their particular system and get it published, right? Because it's a publish or perish system. But we do need to care about it when we're thinking about these large-scale projects. If we want to build things at a scale that is relevant to help us create predictive algorithms that can be used to actually predict how our intervention is going to work, we need to understand bias. Number two, we need absolute quantitative readouts, not relative abundance. The thing that has hampered us most of all in all of these data standards and methodological standards that we've deployed is that we've been working with proportional data. And our proportional data is not useful in the way that we would hope it to be used for things like artificial intelligence-based machine learning approaches, right? So machine learning being a subset of artificial intelligence approaches do and can use proportional data, but it's not effective in helping us to predict really what's going on in the system. It just provides an artificial observation or prediction of outcomes in the proportionality of that system. And that's a problem. We've started to solve that. We've developed quantitative metagenomics and quantitative metatranscriptomics protocols, and one of those will be coming out in M Systems hopefully in September to demonstrate that it is possible. And we're now validating those protocols on about 50 different environments, from marine systems to soil systems to different animal environments, and using that information to benchmark those protocols effectively so they can be deployed by anybody. And that will hopefully deal with this problem. We need more complete complex benchmarks, right? And a complete microbial strain database. So we can completely change our interpretation of our data based upon what algorithm or what database we use, right? So we need very well benchmarked data sets that we then need to fill out a microbial strain database to actually help us to interpret those predictive outcomes. That effort, and I've seen some of those today. I saw the ATCC talking about efforts to get that microbial strain database up and running and get genomes out there, is going to be absolutely essential, but it needs to be done in a way that enables us to intersect with the research community effectively. We need to give up on a microbial reference range approach and understand why they fail. Reference range approaches don't work because the same microbes that make a difference between health and disease, for example, in a mangrove swamp or in my intestine, are the ones that make the difference between healthy in two different places, right? So if we're trying to predict why a system is broken based upon proportional data with a microbiome database, we can't be using these microbial reference ranges to really say, yeah, if you don't have enough Bacteroides, you're going to die, man. That's not good, right? That's just not effective. We need a much more effective way of classifying, say, why sick people or sick environments are healthy and why many healthy people are sick. We can't use these study sites. And we're probably measuring the wrong thing, right? Because we don't even know how to assemble the puzzle pieces yet. So measuring the wrong thing is important because assembling the microbes, the genes, and metabolites into a picture of function is really, really hard. We don't know the syntrophy, the cross-feeding between most of the organisms in the human intestine. And I would argue that despite all of our work in marine systems, the gut is still probably one of the most understood environments on the planet at the moment, and we still know basically nothing. So it's a real problem, right? It's like looking at a fraction of the pixels on your screen is black, and we won't know, we won't help you figure out whether you're reading a book or writing one, because that system is not going to help us to really understand what's actually happening. Okay, so the challenge can be accessed here. So lots of different challenges, from data generation to standardizing those approaches, computational analysis, and I've had some really interesting lectures over the last few days, either remote, either in person, kind of remotely, or listen to in the recordings, and then open access data, so putting it out there and depositing it into public repositories like Cheetah. Reproducibility through different approaches and different platforms like Chime2. Obviously, we're biased to Chime2 because Rob's just over there. And biological robustness, scalability, multi-omics correlation constrained analysis that hasn't been solved, batch effects for processing samples. Remember the EMP, that was well, it still is ongoing, but it's been going for 12 years, and obviously we've sequenced things time and time again, but not the entire catalog, so there's going to be batch effects there. And meta-analysis, and I'll mention red biome in a bit. So every step in this workflow, from sample to result, involves choices that can have large effects. And so, for example, bioinformatics can have large effects. Yes, you know, we've got to get our DNA extraction protocols right. We've got to get our amplicon sequencing and our metagenomic protocols right. But bioinformatics can affect everything massively. The way you analyze it can affect how you interpret it. If you know how to use the methodologies that you're employing, it can invariably help you to understand what's going on. If you don't, it can result in really bizarre findings. The EMP protocols can be done reproducibly by many labs from written protocols, although some still screw them up, so you've always got to check. But developing those methodological protocols was very hard. Developing bioinformatic protocols will be equally as hard. And so the MBQC project was an attempt to standardize them by the functional laboratory-based protocols, and it did a reasonably good job, as I say, but it really wasn't getting at the main problem of how do we normalize the bioinformatic approaches effectively. So these are, for example, some of the methods for profiling in bioinformatics, and they're all appropriate. They all use different approaches, and we get different kind of aligners, for example, but they're all biasing our analysis in different ways. And if you compare the same data set using all of them, you get pretty different results, right? Hopefully this is indicative, right? The legend is just, the colors just represent different phyla, and the types of approaches you employ for metagenomic and amplicon sequencing can really bias how you're looking at that sample. So I've highlighted here some other challenges that are not discussed in purple, and you can see that there's a huge number of them. Hopefully you can see that, because I'm joking, it's moving too fast. But the idea is that we haven't only just started to really understand what's going on, and it really does affect how we can actually interpret these data sets effectively. And if anyone wants that full list, please email me. I'm happy to send it to you. Okay, so what do we need to adopt, and what do we need to create? So one study that we've been doing, the Microsetta initiative, this is our Microsetta initiative local. It's called Microsetta. It's a bit too clever for its own good. This is the Microbiology Rosetta Stone initiative, right? We're trying to create a Rosetta Stone for microbiology. And one very clever student came up with the idea of Microsetta. But we're trying to do this in order to collect diverse microbiomes from their constituent strains using standardized protocols, and use this to understand dynamic shifts. And one of those, for example, is working with Gloria Dominguez-Bello and Marty Blazer to understand how we can create culture collections of all the microbial diversity in the current world, so that if we do lose microbes, and I know Marty's big on missing microbes, but if we do miss our microbes, we can add them back in effectively. And so these study designs, creating standardized protocols and standardized data reporting initiatives and methodological approaches, are really important to be able to do that effectively. We know that taxonomy is incredibly important for standardizing. And if you look at LLPSN, so the list of prokaryotic names standing in nomenclature, they define taxonomy and nomenclature hierarchy in a really, really well-structured way, and it's continually updated. We should adopt this refined standard rather than trying to recreate it, right? We don't need to build something that already exists, right? So we should work with the international community to ensure that we are using these approaches effectively. And we need to be able to match taxonomy to sequence reads, and that to a certain extent does need building, right? We can annotate sequences for amplicon reads and metagenomics and metatranscriptomic reads, and then we can assign them theoretically to a database with LPSN taxonomy and match those references. Currently, those taxonomy databases only provide an approximate mapping, and that requires understanding phylogeny. So phylogeny enables us to interpret those relationships and predict the locations of the unknown taxa by understanding those evolutionary dynamics. The GTDB can do this, right? And you should definitely look that up if you don't know what it is. But we can also do this with the tree of life, and we have started to explore that and with 6NS phylogeny. Using a recently published database on about 10,000 different genomes in the system. Phylogenomics of those domains provides us with a really robust phylogeny. And we can build this into the web of life, which allows us to identify the position of extant and potentially predicted ancestors and the horizontal flow of different gene structures. But this tree is just a start, right? We need this network of horizontal gene transfer to really understand how new genomes are cropping up and to use that information to be able to drive predictive algorithms that can enable us to understand what's going on in your body or this ocean. I say that because I'm actually looking at the ocean right now. Very enviable position being at Scripps Institution of Oceanography. So if I want to predict a unique environment, I need to be able to do that effectively. And then, of course, we get to function, right? So taxonomy, phylogeny, function. Curating the pathways from the literature, very expensive, very error prone unless language prediction approaches can be used with artificial intelligence, which still don't work very well. We can use the existing functional pathways like KEGG, which we use a lot, MetaPsych, Eggnog, TigerFam. There's a good opportunity for machine learning to integrate and predict structure updates for these databases. And that's actually being employed in a number of different groups. And we can infer the functions by associating with different environment and phenotypes. So with things like the EMP standards, which are applied to cheetah, and then using things like red biome, we can take our 500,000 public databases in cheetah and start to say, hey, I found this unrecognized gene. This unrecognized gene appears to be found in relatively high abundance in hydrothermal vents and hot springs, and in this particular, 60 degrees centigrade culture vessel that someone was running in the Netherlands. That's really interesting. Well, this must be a heat-adapted organism. But we can do that because we've standardized all the reporting and standardized all the data, and we know this organism is cropping up. Its functional traits, its taxonomy, and its phylogeny map across this complex series of databases. The reference sample sets are needed to test these procedures. And changing DNA extraction or reference genome database to improve the ability to classify can only be identified if we have solid reference sample sets. So for example, I've heard a number of people talking about the commercially available ones and new ones that are coming out, and we're really excited to see what happens there. We'd like more complex ones, not just one with like 15 different organisms, but one with 200. And really well-defined ones that can produce reliable, predictable outcomes. So we need also gold standard data sets for multiomics. The EMP 500 study, which is available on bioRxiv, but is coming out soon, is metagenomics and metabolomics on 500 different environmental samples, including human, but marine, sediment, freshwater, soil, plants, etc., animals. We need a human equivalent of that kind of study that's more detailed, right, with quantitative data. So applying quantitative metagenomics, metatranscriptomics, and metabolomics to really understand how human approaches are varying and match those to strain collections. Because otherwise, how are we going to know if our approaches are changing the outcome effectively? So as I say, give me a 400 strain representative human gut gold standard data set, analyze that and reanalyze it and analyze it again with all the different approaches and determine which ones are producing variation. So to get at this in a more effective way, we need to standardize even further with robotics. For years, we've been just using the Bristol stool charts, but now we're starting to collect data out of that Bristol stool chart information in terms of genetics. And we do this through microbiome-wide association studies, like the American gut, which I talked about before, which allow us to compare our sample to tens of thousands of samples, both at the taxonomic level and now at the functional level, to identify the different metabolites produced by these microbes and how they vary across populations. And we also know, however, because most of that data is cross-sectional, that time is really important. And so we're now working to increase the number of time points. And this diagram from one of Eric Alm's papers, working with Sean Gibbons, demonstrates that the number of time points you sample on the x-axis affects the variation in your ability to identify a biomarker in a database. So if you've got one sample, the likelihood you will identify per person, the likelihood you'll identify a particular biomarker in that database varies dramatically. But if you've got more than 10 samples or more than 20, then the variance differs very little. So why is time so important? Because unlike height or weight, or blood pressure and insulin, the microbiome and human genetics are highly dimensional. But unlike even human genetics, the microbiome is also highly dynamic. So you need to have a lot of people studied over a lot of time points to really be able to understand how it's varying. We developed this tool, the Gut Lab, which can be placed in a bathroom, takes a piece of used toilet tissue and extracts, does everything a molecular biology lab would do, extracts the information, and in the future will make sequence libraries for sequencing. But right now it just extracts the DNA and RNA and metabolites and stores them in a standardized format. Basically removing the technicians that would normally do that in my lab. Don't tell them, they might get upset. But we're running these alpha prototypes in clinical trials right now to generate highly reproducible data. Based on the benchmarking approaches, which I can't show here for various reasons, but benchmarking across all the available stool collection and analysis and shipping protocols that are available out there, the Gut Lab has the lowest variability in terms of beta diversity dispersion within samples studied across all of the different collection techniques. So it does exactly what we wanted to do. It removes a lot of bias in shipping to collection and processing. And this has been plugged into things like day 2 programs, where you can take the microbiome data and predict personalized nutrition efforts, and utilizing the Gut Lab for programs like the NIH's Common Fund Nutrition for Precision Health program, which we're currently involved in. We hope to be able to produce tens of millions of metagenomes from tens of thousands of people that are all generated in a completely standardized format using these kinds of technologies. So our current work is to deliver metagenomics and metatranscriptomics data on all these samples using these standardized collection approaches, and then to improve the resolution and sensitivity of microbiome analysis using that standardized data set. So just to finish up, what can the microbiome do for us? Let's develop those diagnostics. So for human stuff, predict disease and complications before they happen. You could say that same for environmental stuff. We've got big changes going on with climate change, and so being able to predict how diseased an environment is kind of key to our success and viability. I'm big on commercializing IP, so that's really important to identify those small molecules, for example, or particular organisms that can be used to treat disease. We want to educate on antibiotic stewardship and best clinical practices, and this can really be aggressively achieved using standardized approaches. And we want to redefine that human environmental nexus, get those critical inflection points identified for translating the research into bedside approaches. And then broaden the clinical trials to introduce microbiome research into high-risk diseases, and create those new technologies and informatics to support the globalization of the diagnostic and therapeutic. Right. I just want to leave you here with John Cryan and Sarkis Mazmanian put this beautiful infographic together for a paper recently, which I think really does talk to the way we need to do this for humans. Let's face it, most of the money for standardization will come from human studies. So you identify causality, you need that normal and healthy gut flora, You've got to figure out what that actually looks like, and quantitative metagenomics is going to help us to get there. We need to understand the effect of lifestyle, use more accurate controls like household controls, understand how the genetics and the environment impact the variants in the microbiome, get that inventory of human-associated bacteria, that strain library that we need, all those genomes, and define the directionality between cause and effect, which will actually require large-scale, longitudinal studies so that we can really understand what's going on over time, which can only be really achieved if we have sample collections going on in the bathroom every single day, every single bowel movement, or every single skin sample, or every single saliva sample, so that we can collect them all the time, and the participants can do that at home with no intervention from us. Thank you very much.
Thank you, Jack, for that excellent talk. I thought we could use this as an opportunity for a dialogue or follow-up questions. And I just want to start off saying that it's super exciting to see some of the technological innovations, especially BiomeSense. I'm sure Amazon has you on their radar, given that they acquired iRobot recently. But I think,
I don't know, little robot vacuums might be different to little robot toilet poop samplers. I mean, like it's
sure enough.
Maybe, you never know.
Sure enough. I want to start off with the point that you put out there in the slide in terms of bias, especially from even sampling strategies. Just moving across, from even that space, I think you've been engaged with one other element of equity, which is social equity in human microbiome research. And I think that's a tremendous area to focus on as well, to be included in this bias, because especially as we acquire samples and data sets, and there's a disparity in the geographic regions from where you acquired these data, right? So I mean, there's a lot of efforts that have been happening. And the way some of these efforts have started in the way you want to try to democratize data comes with these challenges. So I think it's a learning curve, and as we move forward, there will be several synergies that can strengthen the case forward. And yeah, I just want to pause there and take those challenges.
I would love to talk about social equity just briefly. I think it's incredibly important. And we have a special collection of papers that have brought together social scientists and political scientists and microbiologists and to understand how social policies and social programs influence our understanding of microbiome-associated diseases. That's in our journal NSystems. I strongly go and look at the NSystems front page, go to articles, look up collections, and you'll see the social equity collection there. It's a really, really powerful collection of papers that will be eye-opening. You have a question? Yes.
Hey, Jack, it's Scott Jackson from NIST. Thanks a lot for the lectures. Very, very nice. You mentioned the need for absolute quantitation, and I believe we would all be in agreement the need for that. How do you envision us getting there? I think, we could all lay out the reasons why we can't do it now or can't do it accurately now. So my question is, how do we get there? What are your thoughts?
So Carsten Zengler, Rob, myself, and a few other people developed a protocol which we're now deploying. Basically, it's really easy, right? It's based on stuff that Mary-Ann Moran did back in 2007 for metatranscriptomic quantification, but it never really was properly validated. And it's been used by the marine environment for a few years, but we've just weaponized it, if you will, for human-based research. But created these 2,000 base pair fragments, which don't hit anything in the EBI or GenBank. We have 10 of them. We then grow them up in E. coli to create absolute cell numbers, which are defined based upon a picoliter printing robot. And then those fragments in absolute cell numbers are extracted in a gradient of cellular abundance from like 1 million down 100,000 down 10,000, 1,000, 100, 1. And then we take those markers and we introduce the DNA into our protocol pipeline for extraction and library preparation. And then we just pick them up using read mapping in the metagenome. And so we then create an internal standard, which has been attempted a number of times and never really been properly validated, but got that internal standard. And then we just read map all of our metagenomic or metatranscriptomic reads against that standard. And what's remarkable is it works. So we take a qPCR of a random subset of genes and check them against this internal standard, and it works really well for saliva, for skin, and now we're benchmarking. We haven't published this yet. Benchmarking it for stool for the Nutrition for Precision Health program. So part of our commitment to the NIH NPH, Nutrition Precision Health. Well, can't say that fast. Nutrition for Precision Health. My mother would say, you have to articulate, boy. But the piece there is really important because we promised to provide that for metagenomics and metatranscriptomics for stool. And so we benchmarked it in a reasonably cost-effective but reasonably well-distributed sample set. And we're going to broaden it out to the 12 or 13,000 people that we're recruiting next year for that program. So we're really excited about that. Everything takes time, everything takes money, but really excited about the opportunity to deliver that. Again, it's bias. It's not perfect, but it's the next step in this evolution towards absolute abundance measures, which I hope will come with a new technology, right? I don't think retrofitting old technologies to this problem is a final solution, but it's our solution right now.
Yeah, that sounds great. So in the case of a fecal sample, you've got cohorts of fecal samples. Would you then say that there are like 10 to the 4th Escherichia coli per gram of feces? So you would have to, even absolute, you still have to, relative to something, per gram of feces, per...
But it's per gram of feces. And so we can even quantify, we've been able to assess that based upon water content, so with a diarrhea sample or a hard pellet sample, we can assess that variance. But yeah, we have to know the exact mass of the sample that's being collected and extracted. That's really important. So with the gut lab, for example, where we're taking a piece of smeared fecal matter on a toilet tissue, it weighs the toilet tissue. So we need to know the average weight of the piece of toilet tissue. So we actually provide that with the device. Here's the toilet tissue we want you to use. Not because there's a problem with extraction, but because we need to know the average mass of these pieces of toilet tissue and that the variance in the mass within a batch is less than the variance in tolerance limits for the prediction of the mass of the stool sample that's collected on that material. It gets complicated, but we're really excited by the potential of using that information to drive quantitative approaches through. But remember, it's not the E. coli, it's these 200 base pair fragments.
Right. Okay.
I do like this camera work, by the way, because I can see you sitting in that nice check shirt. It's lovely.
Hi, Jack.
Hello.
Ben Callahan from NC State.
Hey, Ben.
So your talk had this really broad scope, and it made me start thinking about big picture problems. So you kind of had your 5 points. And of course, some of them are very close to my heart. I care a lot about bias. I think that's maybe one of the main themes of this conference. But one of the big things that we're missing, if we want to understand microbiomes, predict how they'll act, is the biome part, the environment. And I guess there's like 2 really important things that I think we're generally missing. One is usually almost any description of the environment. I mean, there's usually not even, sometimes we'll have like pH or something, but very crude, maybe just what's being studied, but too little to know. Microbes will act very differently in different pHs. And the other is the spatial structure that these guys live in. So when people try and do modeling, almost all of those models are some kind of like pan-mictic mixture where everything runs into each other equally. And so I wondered, these are real big picture questions. So how can we do better on that part?
Yeah, you've hit upon a really big problem. With the EMP, for example, right? In the EMP, we couldn't standardize the metadata collection for every sample. So we asked people for basic minimums like temperature, lat long, time. We got pH for nearly everything because that's reasonably easy to deploy. And then what we were missing, we got salinity for virtually everything, right? What we were missing was nitrate, nitrite, phosphorus, phosphate, silicate. We didn't have any of the biochemical measurements. We had them sporadically, really rich databases, really poor databases, and that causes a big problem. And then when we switch from our unique ability to really easily predict how microbial dynamics occur in water, as you say, really well admixed populations, taking that into, say, soil or mangrove swamps or sediment becomes really, really difficult because of the sparsity of the biology. You got 20,000 cells sitting in a small biofilm on one grain, and there's nothing else on the grain. And then there's nothing else in the area apart from something 50 microns away on this other grain. It's a mess. And so you have to average, right, if you're going to derive predictive capability in that space. But how do you solve that problem? I don't know, man. Ben, you've been working on this. You can do it. Come on, like it will work. We'll figure it out. Don't lose heart. I think the key thing is get down to that functional causative relationships between the organisms. If we can understand syntrophy, if we can understand response to localized environmental parameters, and if we can get better at probing those parameters at a level which the microbes experience, will be great. But I've been saying that for 15 years. When I was younger and more hopeful, I thought we could do that in about 10 years. I thought we'd develop, very, very fine scale probes for phosphate, for example, and we just don't have them still. So I think there's a need to do it. There needs to be a big push in industry to make sure that there is a commercial viability for those products.
I don't know what the solution is, but what I would love is a new technology that's like metagenomics, except for all these environmental factors. Just like metagenomics can sample all the potential microbial diversity, like one measurement that measures all these relevant, chemical or whatever factors at once. So if anyone in industry wants to develop that, that would be really cool.
Yeah, that'd be super cool. We'd love you forever. I mean, it's funny. Peter talks about that with regards to metabolomics, but it's not there, right? You can use untargeted metabolomics to do so much, but it's never going to replace our ability to do physicochemical measurements effectively. Not yet.
Any more questions?
Hi, Jack. Thanks so much. Liliana Brown from NIAID. Very happy to see you, even at remote. Yeah. And we get to see the reflection of the ocean from the glass.
I don't dare turn my camera around, or people get really upset, especially when they're unfair.
Unlike the Earth Microbiome Project, we have many different studies that have collected human samples across using many different kinds of approaches. You see a future in which the same approach is being used across, or the alternate where all different approaches are used, but we can really use the data across the different approaches?
Oh, man, that's a really tough question. Do I see a future where we can use it across all the different approaches? Yes, I do. I think the key thing is, it's really weird, but now you've gone and sat down because it's focused on an empty microphone. I think it's, to me, thank you. No, you don't need to come back. It's fine. It's fine.
It's a question for all as well.
Yeah,
so
I think it's a good.
Camera can...
I think, yeah, from what we need is something which would enable us to analyze as many different variables in a human population as possible. But yeah, I think we need a panel on that, because I think there's a huge number of issues in dealing with that kind of problem.
Not going to be an empty microphone for a while.
Hey, Jack. Scott Ty from the University of Vermont. How are you?
Hey, Scott.
Thanks for the great talk. Always fun to listen to you. So I have this paradox. I see many people, and I've harped on this before, a lot of people spend their whole lives working on just DNA. And I think we're missing some stuff, and I just want to get your thoughts on this, is that we're losing the element of microscopy, seeing as believing, and actually culturing, right? So many of us are just extracting DNA. We don't culture anything. We don't look at it under a microscope. And I just kind of wanted to get your take on it, because I think that has to come back into the picture, and we have to go back into the world of phenotyping at some level, because we could be missing a whole bunch of stuff. I know when we do it with Extreme, we end up almost everything that we culture, we don't even see in our DNA extraction data, and we beat on that to death to try and get everything. And I just want to get your thoughts on that.
Yeah, I mean, it's really important, right? So I would argue that nobody's ignoring them. I've been talking about data standards and large-scale data generation, but no one's ignoring that. I mentioned many times strain collections, and that will only come through culturing, right? And so new efforts to culture all of the organisms in all samples is absolutely essential. And I mentioned Carsten Zengler, who worked with closely here. He's really pushing on that heavily, and we have number of different commercial platforms now available that allow for individual cell or individual micro community growth to allow for syntrophy to support cultivation of novel strains. And that's really being pushed forward. Now, can we keep them alive and in growth culture? No, not usually. But we can grow them up to the point where we can genome sequence them or run a transcriptome and get some more data that's resolved to that one cell or that one sub culture, sub colony, right? Yeah. And then for the microscopy piece, yeah, I would never move away. I mean, Jessica Mark Welsh, who I work with at the MBL, really pushed forward things like classy fish. And classy fish really did enable us to see syntrophic relationships between organisms under the microscope. So in oral plaque, that was really key, and that's still going on. We've used that in animals to study how bacteria on different subsections of the gut interface with immune cells through metabolic interaction. So, it's all very well saying, well, the bugs are producing a lot of short-chain fatty acid. Great. Where are they doing it? Do they have syntrophic partners which will enable it? And how close do they have to be to have an impact upon, say, T-cell regulation? And so using those microscopic approaches with Class C, for instance, in situ hybridization, can really enable us to have another data layer, which gets to what Ben was talking about with regards to structure. The microstructure of that environment is another layer of data which helps us to interpret it. So you're absolutely right, and it's absolutely key to helping us to understand how those systems function.
Yeah, I was thinking when Ben brought that up, I'm immediately thinking about complex biofilms. We don't capture that. And
yeah,
that's
it's funny, like I say we do, but a lot of it gets forgotten, right? So like some of the early metagenomes were in cross sections of complex biofilms in crusted mats around hydrothermal vents. Thinking of the work by Gene Tyson, for example, with many of the teams up at Berkeley and LBNL, various other places. That was really starting to say, well, can I get site-resolved material? I mean, that's like 15 years old now. People do get hung up in the sheer volume of studies that are being published, but there is work out there that we can do. It's just a lot harder. People don't like doing it at scale. And this comes to, what someone was saying, I think it might have been Ben as well, but can we produce these tools to help us get at it, right? And if we can at a large scale, then we will use them. I think everyone's demonstrated that with sequencing. You build it, we will come, right? This whole Kevin Costner fantasy. If you give me a tool that will enable me to investigate things at that scale, I will use it, right? I just don't have the engineering expertise to build it myself.
Thank you, Jack.
Take a moment to thank Jack again for the excellent talk. Thanks again. Thanks, Jack.
Thank you. Wish you could be there.
All right, well, that brings us to the end of day 2. Thank you all for spanning the full 2 days so far. We've still got another full day tomorrow. So one thing I want to leave you with, Stephanie and I are going to put together a Slido poll tonight, and we're going to ask for constructive feedback about things in the workshop that have worked and not worked so well. One piece of constructive feedback that we got at lunch is that some speakers, despite staying within their speaking time frame, didn't have the opportunity to answer questions because we were running late, and it's not really fair to them because they stayed within their time constraints. So these are things that we can improve in the future. If you have constructive thoughts like that, we'd love to hear them. We're also going to give away a prize, a coffee mug, based on some criteria that we haven't figured out yet. Maybe a Slido poll where you all vote on the best talk, and we give the coffee mug to the person who has voted to have the best talk. An idea for yeah. All right, so we have all right, we might have a coffee mug being donated here. Steph, anything else? So we'll start at 8:15 tomorrow morning, right? And no planned activities tonight. Feel free to do your own thing. For those of us who are introverted, we'll go home and sit in the corner. All right, thanks everyone.